Good afternoon, ladies and gentlemen, and welcome by this lecture about the pros and cons of methods to characterize extracellular vesicles. In this lecture, here you see the outline. I would like to cover what do you want to characterize, but also what do you need to characterize. And once we have figured out that, what this means for single EV characterization methods and also for bulk EV characterization methods. Now, we have seen this already several times today, and most of you are probably familiar with this. The ISEV requirements, the first paper on that was published in 2014, followed by one in 2018. But there are other ways to ensure that you're doing the right thing, that you're doing standardized work. For example, EV-track, methodological guidelines to study extracellular vesicles, and more, and more, and more, and more, and more, and more. And at a certain moment, you cannot see the woods for the trees, right? And at that moment, you have to stop and think and realize actually what you want to do. So what do you all want to characterize? I think that's not a very hard question. I expected you all would yell something like extracellular vesicles, of course. But yeah, then you have to take one step down and check what extracellular vesicles actually are. And luckily, like Rink Newland already said this morning, we have a definition now. Extracellular vesicles are particles naturally released from the cell that are delimited by a lipid bilayer and cannot replicate, which means they do not contain a functional nucleus. For detecting and characterization EVs, this means that these aspects we need to measure. We need to ensure, or make it at least very likely, that particles are naturally released from the cell. We need to make sure that the particles we measure are delimited by a lipid bilayer. And we need to make sure that these particles do not contain a functional nucleus. And only if we have confirmed these three aspects, we know we are dealing with vesicles. So, what properties of EVs do you want to measure? The answer should be these three properties. And actually, these you don't want to measure. If you claim you're measuring EVs, you need to measure these. Now, what do I mean with that? Particles naturally released from the cells, how could you prove that? For example, you could measure the size distribution. If your sample also contains cells, but the size of the particles you are measuring is way below the cellular size range, that is forms one clue that you're dealing with vesicles, not with cells. You could measure the dynamics of EV release, for example, with total internal reflection fluorescence microscopy. You could use indirect evidence, for example, the presence of proteins involved in biogenesis. For the delimited by a lipid bilayer criterion, you could use the susceptibility to lysis, or bilayer thickness measurements, or presence of transmembrane proteins to show that your particles have a bilayer. And do not contain a functional nucleus, could be measured also indirectly. For example, by measuring the size distributions. If your particles are smaller than a micrometer and your cells are from mammal origin, you're certainly not dealing with particles that contain a nucleus, or perhaps the DNA concentration. So this means that just applying rules without thinking is not a very wise thing to do if you're dealing with vesicles. For example, characterization of single vesicles use two different but complementary techniques. It's a phrase that contains a lot of wisdom, but for sure it does not guarantee you that the particles you study are vesicles. And I have one example of that. For example, you have some flow cytometry data. Here you see the side scatter versus forward scatter plot. Every dot is an EV. If the color increases, there are more EVs of that combination of signals. And now you want to verify that these particles are EVs. So in addition, you do a transmission electron microscopy image, and you conclude, okay, the particles I'm looking at are EVs. But that conclusion is not necessarily true. Why not? You need to calibrate and you need to quantitate. And if you do so, you will see that in this case, and this is the same data, the electron microscopy and flow cytometry data are measured in entirely different size ranges and entirely different concentration ranges. So here in blue, you see the size distribution quantitated with electron microscopy, and in black, the size distribution of particles measured with flow cytometry after calibration. And so these techniques do not confirm that you're looking at the same particles. So what properties of EVs do you want to measure? So first, make it likely that the particles you're dealing with are EVs, and then you can start measuring the property of interest, like antigen expression, concentration of EVs, expressing certain antigens, total concentration of microRNA, etc. And if I walk around here at ISEV, I have the feeling that's the key capacity of many labs. Maybe it's even the core work of your lab. But once you have to make it likely that the particles you're dealing with are EVs, and the signals you measure originate from EVs, it's getting harder, right? Now, with all that knowledge in mind, let's see how we can project this to single EV characterization methods, such as electron microscopy, flow cytometry, nanoparticle tracking, or resistive pulse sensing. So here, let's start with cryo-electron microscopy. So we have to confirm that we are dealing with particles naturally released from cells, delimited by a phospholipid bilayer, and absence of a functional nucleus. Now I think we can readily say that we are not dealing with cells, at least. So the size is an indirect evidence, could be an EV. With cryo-electron microscopy, you make an image of the bilayer. You can see that clearly here. And due to the size, a nucleus doesn't fit in such a small particle. So you can fairly state we are dealing with vesicles here. With flow cytometry, we cannot directly make it hard that particles are released by the cell, but by a calibration, we can at least do the sizing and confirm that we are dealing with submicrometer particles, that they are delimited by a phospholipid bilayer. There are several ways for that. For example, antibody staining. If the proteins are transmembrane proteins, then they have to be in a phospholipid bilayer. Or we could do lysis or staining of phospholipid bilayer directly. And the absence of a functional nucleus, we could get this indirect evidence from the size. TURF microscopy. So I got this nice video here from Guillaume. Unfortunately, it doesn't start. But it's a technology where you can directly image the EV release by a change in fluorescence, or by a change in pH that makes the particles fluorescent. And so yes, it's very clear that these particles we measure are released from the cells. It's a bit harder to confirm that they have a phospholipid bilayer, but the size of these particles, they are subdiffraction limited, means a nucleus wouldn't fit. And so you could do this for any technique, and yeah, for the sake of time, I'm not going to do that. I added just 3 more. And please don't think that if one of the cells here is green, that I state that that's a good or a high quality technique. No, this table does not summarize the accuracy and quality of methods to determine the size and antigen expression. For that, I would like to refer you to the literature. So this table shows only whether we can confirm the 3 aspects, the 3 properties that make a particle an EV with these techniques. We could do the same, of course, for bulk EV characterization methods, such as an ELISA, omics, or Western blot. And the picture would look a little bit different, because with bulk EV characterization methods, it's very hard to prove that you are dealing with particles that are naturally released from cells. You don't have any evidence for that directly from the signals of these techniques. You have indirect evidence that you're dealing with particles that have a phospholipid bilayer, for example, via antibody signals that you can pick up. If the antibodies target an antigen that is transmembrane, then these particles had or still have a phospholipid bilayer. And also it's generally hard to confirm that these particles don't have a nucleus, except maybe from genomics. If the DNA content is very high, that may be evidence for that. It's hard here for these bulk EV characterization methods because signals may originate from other particles in your sample, such as cells, such as soluble proteins, circulating RNAs or DNAs. And I have an example of this. This is because platelet-free plasma typically still contains platelets. So what we did here, we prepared plasma according to an ISTH protocol that's used by many labs. It involves double centrifugation for 15 minutes at 2500 Gs at 20 degrees Celsius. And these were 3 studies done in the lab, in collaboration also with other labs. And in those studies, we measured the concentration of platelets. And as you can see, there are 2 things striking. First, 3 studies using the same protocol resulted in different platelet concentrations in this plasma. Secondly, this platelet concentration is at all not so low. It exceeds at least 100,000 platelets per milliliter. Is this a confounder? Yes, it is. Here you see the vesicle concentration in red for CD61 positive vesicles. Those are vesicles derived from platelets. And in green from CD235A EVs. These are vesicles from red blood cells. Versus the concentration of these residual platelets in plasma. And you see clearly that the concentration of platelet-derived vesicles scales linearly with the presence of platelets in plasma. So for bulk EV characterization methods, confirm that signals originate from EVs. Generally, that requires single EV characterization methods, but it also requires methods that confirm the absence of confounders, such as cell-soluble proteins, circulating DNA, etc. And if you do so, don't forget about the detection limit. And for that, to explain that, I prepared this figure here from the compendium on flow cytometry. You see the concentration of particles versus diameter for extracellular vesicles, but also for lipoprotein particles. So this graph basically reflects the situation in blood plasma. Now, if I would measure these samples with 2 detection techniques, technique 1 and 2, which have a different detection limit, technique 1 would only find vesicles in this example, whereas technique 2 would find vesicles as well as lipoproteins. So this is why it's important to figure out what your detection limit is and report that. And don't only consider the diameter detection limit, there's also a detection limit on concentration. So in this case, technique 2 would only find lipoprotein particles and entirely miss the EVs. So be aware there is a detection limit, figure out where it is, what it is, and report it. One example of that, for example, you want to quantify the purity of EVs in blood plasma relative to cells, so concentration vesicles compared to cells. Your hematology analyzer measures 0 counts. So you would conclude there are no cells, and perhaps you have a 100% pure sample. However, if you would have carefully read the instruction manual of your hematology analyzer, the detection limit is 2 million cells per milliliter, and we now know that yes, it makes sense that the hematology analyzer provides 0 counts, 0 concentration. But everything below the detection limit was missed. So for sure you did not have a 100% pure EV sample with respective to cells. So in sum, confirm that signals originate from EVs. So measure EV properties related to the definition, so that are 3 puppet properties. For that you need single EV characterization methods. Measure other desired properties as well. If you choose to use bulk methods, confirm the absence of co-founders and estimate and report the lower limit of detection. I would like to acknowledge all of my co-workers, and I would like to thank you for your attention.